First up, we have Charles Liu, who is a statistician and software product manager at Cytel. He will give a brief presentation before Pat Mitchell. Take it away, Charles.
Okay, thank you, Nathan. Welcome everybody to the webinar. Thank you for attending. I'm very excited today to be participating with Pat Mitchell in this webinar. And while we're preparing for today's presentation, Pat and I agreed that it might be useful for me to offer a little appetizer before Pat serves the main course. And so I'm going to do a very brief literature review of relevant go-no-go decision methodologies utilizing this dual target method. So what Pat's going to present later on is going to be one specific version within this broader family. I just wanted to give an overview of all of the other proposals that have been made in the literature. So within this methodology, there are a few basic components. So the first is typically a treatment effect, although it could be some other endpoint or variable, but typically it's a treatment effect, which we call delta. And the key to this approach is the use of two different values of the treatment effect. These are the two targets, and they are often labeled as a lower reference value and a target value. So the lower reference value could be a null value or a minimum clinically important difference, depending on the application. And the target value is just a value that's better than the low reference value. And then the final component are the decision rules themselves, which are probabilistic, based on either posterior probabilities within a Bayesian framework or confidence within a frequentist framework that are tied to the two reference values. So why would we want to use two targets? Well, I'm just going to give a quote to this recent paper by Stenis et al., who proposed that the value of a drug is not well represented by a single threshold below which the drug has no value and above which the value proposition is clear. So by adding an additional target or threshold, we can arrive at a three outcome approach, which gives us more flexibility. So my first paper I'm going to show you is by Fish et al. So they use a fully Bayesian approach, and here's one example from their paper. Basically, their go-no-go rule is tied to two thresholds shown here. The first one they label as significance, which implies high confidence or posterior probability that the effect of the drug is better than zero. And the second threshold they label as relevance. So I guess this is clinical relevance, where they want to be moderately confident that the effect is larger than some target, in this case 5%. So if we map that back onto our dual target jargon, the significance criterion corresponds to the LRV, the low reference value, and the relevance criterion to the TV or target value. Now, before Fish, Cartwright et al. Proposed also a dual target approach, which was slightly different. And here, if you allow me to quote them, they say, if there is a sufficient probability that the effect exceeds the target value, there is a clear go decision. Conversely, if there is a sufficient probability that the effect will not exceed the low reference value, there is a clear stop decision. So in this case, the way it differs from the Fish et al. Approach is that the go decision is based solely on the target value, and the stop decision corresponds to a low reference value, whereas in the fish, both the significance and relevance needed to be satisfied. Wiley et al. More recently proposed a fully Bayesian approach that extended that proposed by Cartwright et al., where the go decision was tied to the target value, the stop decision tied to the low reference value. And going back further, this is the third approach I'm going to mention by Lalonde et al. They had an idea that was very similar to what Cartwright had proposed, except it was except they switched the correspondence between go and stop. So the go decisions in the Lalonde approach are tied to the low reference value, whereas the stop decisions are tied to the target value. And this is the approach that was subsequently adopted by Frewer et al. You see Pat Mitchell here in this recent paper by folks at AstraZeneca, where they took this Lalonde approach very seriously and implemented it throughout the organization. So I won't go into too much detail about this. I'm sure Pat will reveal more. And to finish off, just want to give a plug to this more recent paper by Senator et al., which again is an extension of the Lalonde approach to a fully Bayesian framework. So thank you very much for your time. I'm going to now hand it over to Pat. And if you have any questions, please, you can use the Q&A along the chat. I'll be watching your questions, and Pat and I will be available after his presentation.
Thank you very much, Charles. So, as Charles mentioned before, we here at AstraZeneca have kind of taken one of the approaches and have kind of taken that and really kind of made it part of our culture here at AstraZeneca. So that's the one that I'm going to focus on talking about today. So I guess the first thing I wanted to kind of ground people in is something we all really know, and that is that drug development is extremely time-consuming and really expensive. So what we need to do, is to try and find ways to make programs shorter. And, in making them shorter, you want to look for more chances for earlier decisions and more efficient processes to help you interpret data that you get from your experiments. One of the main benefits of having go-no-go criteria up front, I believe, is that you can take some of that time that was initially used for interpreting the results after the experiment was over and kind of shift that to more of a front-loaded approach and so forth. So they're in the design phase. You can imagine thinking about the sample space and where your treatment effect can fall in that, and then kind of partitioning that and mapping that to various decisions, which is what I think all of these things really do. So the first step in doing that, I think, is not really in the decide tool, but it is part of our process, and that is trying to set the, you know what, I made a mistake here. It's trying to set the two targets that we have as inputs for the method. The first one is lower reference value, and the lower reference value is what we think we need to achieve in order for the drug to be at least licensable, if you will. So it's the smallest clinically meaningful treatment effect that we could observe. The next target is something we call the target value. And this one is one that we believe if we achieve this, then we create a drug that people will want to prescribe over something that is already approved. So bearing those in mind, we also put in a couple of other ingredients into this method, and those first two in the slide here are basically decision risks. All right, they're very similar to how we set up, registration trials with type 1 and type 2 error, except that here we're thinking about things in terms of an acceptable risk, which is the probability that the truth is better than the target value, given that you make a stop decision. And here's that counterintuitive link between stop criterion and the target value. And then corresponding to that is the opposite risk that we think about, and that is the desired confidence that the truth is better than the lower reference value, given that you make a go decision. So here it is. Your go decision is now hooked up to the lower reference value. Sample size and variability are also required, and those are a little bit different depending on the variable that you're thinking about trying to use as a decision variable. So you can imagine that, if you're talking about a single arm binomial design, as you see in oncology a lot, that's going to be a different variability than you would if you had a normal variable or something like that. So once you have all that in place, you're set up to actually calculate the go-no-go criteria. Now, there's a couple of ways to kind of do this. I mean, this figure on the right here is an interpretation figure that you may have seen in some of these other papers. I know that we have it certainly in the Lalonde paper. I think it might also be in ours as well. This basically says that if you construct an unbalanced confidence interval and you put it up against your target and your lower reference value at the end of the study, then this is how that result will map to either a green, which is usually a go, an amber, which is usually a consider, or a red, which is usually a stop. Now, I made very clear that this is an unbalanced confidence interval, and that is that the lower limit on that is actually a one-sided 100 times the DCLRV confidence level. So if you're doing a desired confidence of 80%, then your lower limit on this confidence interval will be 80%. Your upper limit, on the other hand, is 100 times 1 minus the ARTV, which was the acceptable risk. So if you're doing, again, 10% ARTV, then the upper limit of this confidence interval that we're talking about is a 90% confidence interval. That will always work, but I think there's an easier way to do this, and I'm going to come to that later when I show you the go-no-go figure that we generate with Decide. So more decisive trials are better. So, if you spend a lot of money and you do an experiment, you want something that is going to tell you or how you can go forward in development. So do you either stop development after that experiment, or do you go on to the next phase? You can clearly get what we call consider results, and these are the ones that we've called amber results internally. These results, if you get an amber result, usually tell you that you either need information from other variables of the trial, or if you're doing this at an interim, maybe you need more sampling to let the trial run longer to give you more information. A decisive trial, in my mind, is something that kind of minimizes that probability that you're going to wind up getting a consider result. And, we've adopted the probability that consider, given either the target value is true or the lower reference value is true, are actually better trials in this framework. So what you're willing to call a sufficiently minimized trial is dependent on your internal conventions. So your governance boards, your companies willing to spend this, spend money to actually get to that next experiment will expect that trials are designed in a certain way to minimize that consider probability. Also, there are plenty of projects out there that have looked at a consider result and said, okay, well, instead of actually getting more information, maybe I want to act in maybe a more conservative way than I would if I observed green. This is also certainly okay, and it depends on the appetite of the organization to take on such plans. So I'm going to move on to kind of demonstrating some of this, not necessarily in a formal demo, but just with some screenshots of the Decide tool. Now, the Decide tool is a piece of software that was developed in partnership between AstraZeneca and Cytel, and it implements the method that I'm describing to you now. So to get to the example, suppose we have a real trial, and it's in early oncology, and many of you will get this, others not so much, but it'll resonate for early oncology. The indication you want to treat at the end is renal cell cancer. You're designing a phase 2A single arm with overall response, which is a binomial variable. Your dose expansion trial has basically just finished, and that itself was an all solids trial. So you got a mix of different cancer types in there. After doing this trial, you're going to go on to this expansion trial. While you led up to that, your group decided that your lower reference value was going to be 20% and your target value was going to be 40%. And actually, I'm going to backtrack a little bit here so that you can see why we decided that. This figure here is basically a forest plot of all the literature across various lines of therapy that helped us set these targets. It's a different subject, but the idea is that, after discussion with cross-functional group and considering these data, this is where that lower reference was set, and then this is where that target was set. So just to shift back to where I was, bear with me, please. So you accepted those targets. The ARTV and DCLRV are set at 10 and 80. Those are something that are set, at least in our company, organizationally. The sample size that we expect to use is an N of 25. So first thing we do is we log into the Decide tool, which is a very familiar process. You put your username in, your password, and you hit log in. And then there's a series of options as to which endpoint you can choose. And for us, because we're talking about a single arm binomial, we're going to use a single proportion. So the next step is to enter the information. Okay, so we go in, we put in the target value of 40, the lower reference of 20. This user interest value here is something that is reserved really for the team to decide, I want to look at a particular true because it has some particular relevance to the project. I have it set here as being in the middle of 20 and 40% because I believe that's where I'm going to wind up observing the highest proportion or the highest probability of getting an amber result. Number of analyses, we'll come back to that later. Sample size, here are your decision risks, and you can see this radio button up here. We have it set in this example, the frequentist. We'll come to the Bayesian part later. Let's see here. Okay, so once we put all that in and we hit the compute button, which I didn't show you, we get a lot of information. And it doesn't come in the way I've got it on the slides. There's actually a nice PDF report you can get out of that. But what I'm going to do is I'm going to kind of go through some of what you get piece by piece. The decision criteria, the most important thing that we want to show the broader team, basically are defined by where the red and amber border are and the green and amber border are on this figure. And what I'm going to do for the purposes of our discussion is just kind of translate that back to a number of responses. So what this says to me is that if I have no more than 6 responses, then I'm going to recommend we stop development. If I have 7 out of 25 people responding, then I'm going to say that we need more information from some of our secondary variables, because this is a final analysis we're talking about here. If we wind up with at least 8 responses out of 25, then I'm going to recommend that we go to the next phase development. The other part that is definitely interesting to people in our discipline, I'm thinking statistics here, is the chance of amber given the various truths that we talk about. So, let's see here. So, the table that comes out of DECIDE is always ordered the same way. So you have target value in your first line, lower reference in your second, and user interest value in the third. And you can see that the chance of amber under all of those truths, even the one that, I kind of put in there as maximum amber, if you want to think about it that way, is less than 20%. That actually is probably pretty good for early development. But ultimately, what kind of tolerances you have, you're going to wind up having to discuss in your organizations. So there's a lot more stuff in the main decision figure. And I'm going to go through that in a little bit more stepwise manner. The first element is the lower reference value. So if you look in the green dotted boxes here, you'll see that the lower reference value is kind of called out explicitly on the top of the figure. That's represented by this dotted vertical line. It's also mentioned explicitly on the right side of this value or criteria description box here, and then it's called out again against the go criteria. So, the next thing I want to point out is the target value, and that is the number also called out on the right side of this figure, represented by a solid line, and it's opposite the stop criterion. Third is the fact that we've also thrown in our ARTV, our acceptable risk, that the target value is, We also have the DCLRV called out against the go criterion appropriately. And then we have the actual go criterion, and that's this little green shaded area here. If you see at least 32%, then you're going to go. I'm also going to point out that that is represented by this borderline between amber and green. And then you have the stop criterion, which is a similar criterion. The other thing that's important to notice is the sample space. Okay, this figure is something that we would generate in planning, and then at the end of the trial, we would come back with this figure, and we would also represent the observed overall response on this, so that people could explicitly see, all right, you are in either the red, amber, or green. That would be the basis for our recommendation. One of the other nice plots that you can get out of DECIDE is this probability of red, amber, green given the true, and it's done over a range of truths. So having this in your back pocket, or being able to show it to people with a very slim strip of amber in there, would give some confidence as to the decisiveness of your trial. So, what if you wanted to make an earlier decision? All right, what if you wanted to stop due to futility? Well, DECIDE can do that as well. And what we're doing is we're now taking that same input screen that I showed you earlier, and we're now changing the number of analyses to something more than one. I chose 2 just because it's easy, but you can have more than one analysis in there. So we're going to choose 2, and it's going to give you this little box here that's kind of faint, but it's below the blue bar there. And the interim type is going to be one of two options. Either it's going to be futility or it's going to be administrative. Futility, we intend to stop the trial if we see results that come in below the stop criterion at the interim. The administrative analysis is kind of a more forward, well, not forward-looking is not the right one, but it's more aggressive, if you will, that allows you to maybe trigger parallel activities. So if you meet the stop criteria, or if you don't meet the stop criterion, then you stop the trial that way. But if you meet the go criterion in an administrative analysis, then you're triggering maybe manufacturing, maybe earlier planning for phase 3 or something like that, depending on how your program is set up. So we choose futility for this, and we're going to add the interim following 40% information. So with a 25 sample size, we're going to do an interim after 10 patients. So Decide then gives you a large amount of other information, and the first of which is what the stopping criterion actually is. So in our example, following 40% information, if you have no more than one response, then you intend to stop that expansion and stop development. This larger table down here illustrates the difference between the risks with the interim and without it. And you can see that under the target, for example, the chance of stopping with the interim is 10%. The chance of stopping, meaning getting a red result here, the chance of stopping without the interim is only 7%. So adding the interim doesn't raise your overall risk by a lot. Now, at this point, I'm going to point out that, this is probably the difference between at least earlier and late phase when thinking about these things. If I'm doing this kind of analysis for a late phase, maybe license driving trial, then my goal is going to be to try and control that overall type 1 alpha at a particular level. 0.05 two-sided is typical. In early, we maybe take on a little bit more risk. So that interim that we put in there might raise our overall risk of stopping it appropriately. But as long as it doesn't raise it by a ton, we're okay with that usually. And that, again, that's probably something that is a company-related thought. So what I'm saying here is not something that can be generalized. The expected sample size under a range of true values is valuable because it basically illustrates, well, how likely is it going to be that we wind up actually making it past the interim if we don't have a compound that actually has decent performance. And as you can see in the graph here, this dotted line here will show you the impact of adding that interim over a range of true response rates. Let's see here. The user interest value, as I mentioned before, being there at maximum amber, only affects the chance of stopping by a few percent in our example here. So what happens then if we were really fortunate in our dose escalation phase? Say that we wound up with 4 patients in our all solids expansion, which is made up of all solid cancer types, that were actually renal cell carcinoma. Well, we take that information. We have 4 patients in renal cell carcinoma. They're at the line of therapy that we want to treat because that's the way we set up our inclusion criteria. They're all at the dose that we want to study because these are all at the maximum tolerated dose or the recommended phase 2 dose, depending on how you select them. 3 of those patients respond in our example. We're very excited about this. And okay, so you can incorporate that information into your design using the Bayesian methodology and Decide. So again, you have your three trues that you want to look at. Right now, we don't have this for interims, so we have it set at one analysis, but that's coming. We have a sample size of 25, our decision risks, and now we switch over to Bayesian and we calculate some prior. Now the numbers here might look a little strange, mainly because we're talking about small samples. So 3 of 4 gets kind of adjusted down to be no higher than 10% or whatever you're comfortable with, 10% of the overall N in the planned expansion. So decide now tells us something a little bit from a different angle, same information. Here you have decision criteria based on posterior probabilities, and it says basically that if I have no more than 6 out of 25, then I'm going to stop, and if I have no more than 7 out of 25, I'm going to continue. Well, okay, so there's nothing between 6 and 7, so why do I have this amber band here? The amber band is owing to the fact that posterior probabilities are calculated as a continuous variable, not so much as a discrete one, even though you can only observe certain values in the sample space. So the other point here is that there's a convention that is embedded in this that says that if your go criterion winds up being lower than your stop criterion, then that automatically gets reversed. And you can see why I'm raising this, and that is this green curve here is the posterior probability that your true is going to be greater than the lower reference value. The red curve, which is not relevant right at the moment, well, it's not relevant. The red curve is the probability that your true is better than the target value. And if I took this literally, and I looked at where the green curve intersected 0.8, which is a DCLRV, you can see that that's slightly below where this red curve intersects the ARTV, and that's what's triggering the convention here, because it doesn't make any sense to have a go criteria that's lower than the stop criteria. In the chance of amber table here, we can see that amber has now gone from less than 20% to 0, mainly because you just can't get anything when you're counting things between 6 and 7. So the tool itself has been very helpful. It has provided a consistent approach throughout the company for all studies. So when you come to governance and you say, please give me money so I can run my program and get to the next phase, all programs in our company have to have this output. The relevant outputs and reports, when you put in all the numbers into Decide and you click compute, save for 1 or 2 cases that require simulation, come out almost instantly. The results can be shared in real time during a meeting. So with instant results, you can change numbers, you can hit compute, and your team can see things change as things are going. So what's next? We don't think that what we have in the tool right now is the end-all, be-all in terms of everything that you would necessarily want in terms of trying to set go-no-go criteria. So we have kind of collected some feedback on the tool and are working with Cytel currently to try and expand the functionality. So I think that this will continue, for at least as long as I can say anyway. So here are some references. Some of those I think Charles referenced before. There are a few others that take some other approaches and stuff like that. If you look for just go no go, I've pulled out 3 here because I've probably quoted or they're heavily based on what we're talking about there. And then I have my email address here as well. Please feel free to send me method questions about this. All answers are free, and thank you very much for listening to me this last few minutes.
Hello, and thank you, Pat. This is Mike White, Director of Marketing here at Cytel, and together with Charles Liu, who began today's presentation. And before we get into the question and answer period, just to remind people that you can pose your questions to Pat using the Q&A panel. We'll try to get through as many questions as time allows. We do want to thank everyone for attending today and listening. This is, of course, part of our continuing series of trial innovation webinars. And Pat has been so kind to share his work and the work with his colleagues at AstraZeneca. Pat, maybe to get things kicked off with the questions and answers, we talked about this earlier. A lot of times, many people have expressed to you and Charles before about your thoughts about extending this methodology to include multiple endpoints. Maybe you can speak a bit on that.
Yeah, that is some of the feedback that we got in early versions of this. Right now, we don't actually have a mechanism that simultaneously allows you to look at, say, maybe a primary safety and a primary efficacy at one time. But what I have seen projects do is you kind of take those two variables separately and take their outcomes, their red, amber, green outcomes, and kind of cross them into a matrix, so to speak. And you look at each of the cells in your planning discussions, and you go, all right, well, if I see green in efficacy and maybe amber in safety, we're going to do one thing. If I see another combination, we're going to do another, and so forth. I've seen that. I've also seen where projects have taken, say, the main efficacy and maybe treated it in terms of like a step-down approach or something like that. So, give me your main efficacy. If that gives me a clear decision, great. If it's amber, then go to the first secondary or something like that. So there's a lot of ways that you can creatively put these things together to fit your project.
And Pat, we're starting to get some good questions here coming in. So we'll be addressing folks' questions real soon here.
Okay.
But also, some people are asking about moving beyond just the very earliest of studies. What are your thoughts about moving, the framework and applying the framework to late-stage trials as well? And that would be both, on the Bayesian approach and the frequentist framework.
Okay. I think that there is space to apply this in late phase. Now, when I say that, I'm not thinking about applying go-no-go criteria to trials that directly affect licensing or anything like that, because really we already have something that does that. Basically, it's the P.05 two-sided that tells you whether you have something that's submittable. But what I'm thinking about is using that licensing trial and then thinking about whether or not you're going to branch off into life cycle management trials. And when I think about this, I go back to my experiences with Berlinta in the Plato study. Now, for those of you who don't know, the Plato study was a very large outcome study with 18,625 patients. And it was in a cardiovascular endpoint and so forth. But with such a large study, there's a lot of potential to want to look at various subgroups, things like stroke, PAD, etc. And what you can do as you're kind of doing your other things, waiting for this trial to read out, you can do the research in an indication like stroke or PAD or whatever it is, get that forest plot, set your criteria, and then apply a particular. You can apply the the anticipated size of the subgroup that you have in your trial as the N. And that will give you go, no go criteria so that when the large study reads out, you can go and do your subset analyses and look at whether you are in a reasonable red, amber, green range. And you can then go and say, all right, well, I want to do this and I don't want to do that, and so forth. So I think that not only can you apply it there, but you could probably even apply it earlier in development. I mean, even in the preclinical areas and so forth, depending on how you set those criteria.
Thank you, Pat. Among the many excellent questions we're receiving is from Dick Bittman, and Dick asks, for the Bayesian application in the presentation, the prior data were from an AZ study. Now, in other applications, have you found it useful to take the Bayesian approach using published historical results?
I don't think that I have an example of having done that yet, but if there is enough evidence of representativeness, then I think that you are just fine to do that. We have some guidelines as to what we look for internally, and those may be something that is either agreed with or not agreed with as you go from company to company and so forth. But I think that as long as the assumptions that you carry with you when you look at historical data to data that you're about to collect are met, then I think you should be just fine.
We got an interesting question here from Peter Coleman. Peter asks, what would you say are the pros and cons of the Lalonde et al. Approach compared to the other methods by Wally et al.?
By who again?
Wally.
That's not one of the ones that we covered today, right?
Yeah, it's one that Charles, in his reference presentation to start with.
Oh, it isn't Charles.
I guess the question is, what is it about the Lalonde method that AZ found particularly well suited?
Right. Well, that's a question that might be a little bit before my time with the method. Given my very cursory look at the methods that Charles presented, one thing that I would explore is the relative size of the amber zones. When I looked at what we were getting in the literature search, my impression, right or wrong at this point, is that perhaps some of those methods are leading to maybe wider amber zones than what we're seeing here in the Lalonde method when you consider taking the same target and lower reference value. Now, I say that at a very, very high level. I have not done any work in that, but I think that one thing that I will do is kind of look those over and see what we can actually find. That's probably the first thing that would come to mind.
I'm back. Okay. Pat, can you hear me okay?
Yeah, just fine.
Yeah. Sorry, we had a little technical glitch there.
That's okay.
We're going to ask you a question from, I believe the name is, forgive me if I mispronounce, Nilani Lianej, perhaps. The question is, can you please explain a bit more about how you set the DC LRV and ARTV rules?
Yeah, and let me quickly go back to that rather small diagram, and I apologize for the size on that. Am I still? Are you guys able to still see my screen or no?
Yes, we can.
Okay, good. So I won't futz with that then. All right. So this diagram here, and there's really two ways that I've seen it done. First way is if there's plenty of information out there, if you have a lot of published trials. Then what
Hi, Pat. Sorry to interrupt. Just pass you the ball if you want to go ahead and share that screen.
Oh, I'm not sharing. Okay. Yeah. Oh, sorry. Actually, this is okay. Let me. put the figure back where it was because I had my own technical issues. Let's see here. Bear with me a minute. I'm sorry about this. Why is that? Resume slideshow. I guess maybe it's this way. All right, maybe it's here. Maybe it's this way. Aha. Okay, that's why it was. That's why I couldn't get it before. Sorry for all my mumbling here. I apologize. What I'm showing you here in a relatively small diagram is a forest plot that has all the results that I could find from renal cell carcinoma across a number of lines of therapy. And the process by which we look at these data and then set either a lower reference value or a target value is very much a balance in thought process between biostatistics and medicine. And, it's kind of informed by the data, but it's very much very much a feeling and so forth at this point. There's no math behind it other than what you see in the figure. Now you could do a meta-analysis or something like that and actually kind of zero in on very relevant studies and come up with a range of treatment effects there. You could use that as the basis for starting the discussion. But my impression is that something that leans entirely mathematically is more the beginning of the discussion rather than the end of it. And that's as true with the target value as it is with the lower reference value. And one of the things that I've noticed is that in the discussions of trying to set these targets, people oftentimes, without really realizing it, kind of slip gears from the question of what do you need to see in terms of to be successful into what do I think my drug can achieve. And that those are very different questions. One is a matter of target setting. One is a matter of probability of success. We're not talking about probability of success in any of this right now. So if you have the benefit of a lot of data, then a figure like this is terrific to put together, and you have something to talk about. Now, the opposite case is where you have almost no data or no data at all. And this is something that happens in oncology a lot because with 200 tumor types and an uncounted number of subtypes by biomarkers and so forth, not everybody has had a chance to go and actually test drugs in all of those things. So you may have a scientific basis for going into a particular segment, but there aren't any data there. So now it becomes a little bit less mathematical than even putting a forest plot together and so forth. I mean, you can start with a forest plot, but then you have a conversation that, kind of goes, all right, well, here are my upward forces, here are my downward forces, and we wind up in a particular place. Ultimately, in AstraZeneca, we take this forward and we actually agree the targets not just within the team, but with governance. And there have been a number of cases where, we've had the robust discussion and governance has come back and said, well, wait a minute, we think you need to adjust them up or down or something like that. It's really not that hard to do. But right now, I think that setting targets and maybe even calibrating people is an open area in this area of statistics.
And Pat, thank you. It's interesting because your example here, you made reference to renal cell carcinoma.
Mm-hmm.
Once again, when we're giving examples in oncology, a question that often occurs is how would the approach fare in other therapeutic areas? Have you, AstraZeneca, actually engaged beyond oncology?
Oh, yes. This is something that's early development wide. So, the big TAs in our company, respiratory, cardiovascular, they do the same process. So they have to go through the same evidence gathering methods to actually set criteria to get something agreed in governance and so forth. It's very much part of the culture, company wide, really. Does that help?
Yes, it does. It does,
Okay,
absolutely. Lisa Chen has had a couple of interesting questions. Let me see if I can combine them for you. Her first question was around, what are your thoughts about expanding the framework to also cover phase 1B studies with multiple doses? That's part one of her question. If you just give me a second, we'll see the other part of her question. Stand by, please. So Lisa also asked, what are the main considerations for using the tool in phase 2, other than type 1 error control? Did you get that?
Okay. Yeah. Okay. So the first question, multiple doses. To me, each dose you give, patients really kind of creates like a subpopulation. So, I'm thinking about what happens in the dose response curve and so forth. So, if you believe that with increasing dose, your effectiveness rises or your safety rises and so forth, then the true that's associated with a particular dose, and let's keep it in efficacy land, so to speak, that is going to be lower. The response rate is going to wind up being lower than with a higher dose. That said, the targets that you set are really not, in my view, either drug or dose dependent. They're actually attached to the disease. So, when you go and you put these forest plots together, say, you're actually asking a question that goes something like, what does my drug need to do to be successful in the market? And by drug, you can think drug/dose. So if you were to run those two doses in a trial and look at each under its own merits, with that assumption that I kind of laid out before, you can imagine that the drug with the lower dose is going to wind up having fewer responders. So it's probably not going to succeed as often as the one with the higher dose. And again, I'm really overgeneralizing here. But the bars, the hurdles, are the same because both of those drugs at whatever dose treat the same disease. And as far as the errors that you worry about, at least in my experience in early, the concern is not so much whether you are detecting any signal, let's say, like signal different than the null, which is more a phase 3 facing concern with some small probability. The concern in my mind with this is whether or not you send a drug to the next phase of development in error, or whether you stop a drug from going to the next phase of development in error. So those are a little bit different concerns, and they're not really anchored to the traditional null. And the reason why I say that is that if I compare to if I did a placebo trial in phase 3, then my null is going to be no difference, more or less. But if there's an active drug out there, then I necessarily have to beat that drug. And my concern ahead of being able to design a trial that goes head to head with that drug is, well, how much more then do I have to beat either a placebo trial in early phase to actually kind of have confidence that I'm going to wind up succeeding versus the active in phase 3. This is the kind of thought process that we kind of bring when we're thinking about this particular method. And actually, I don't even think about this in terms of like a null and an alternative. I think about this in terms of a double alternative, really. I hope that helps.
I like to think a double alternative can only help. Pat.
Yeah.
So we're at the top of the hour, and we have another good question here that we're likely going to conclude with today. But before I do, I want to thank you, Pat, for your presentation and your excellent work and how well you communicate and share the work with others. That's precisely what we hope to do here in these webinars. Yeah. And of course, thank everyone who's joined us today. Reminder that you will see by Monday an email from me once again with the replay link. Of course, the slides, are on our website right now at cytel.com/event. We certainly hope that you share those with your colleagues, both within your organization and those in your network. That's what why we do these things. And Pat, I also want to thank, of course, Charles Liu for his presentation and framing today's talk with the references. So Catherine Speed, Pat, she asked a pretty basic question. How long has AstraZeneca been using the Decide software? And then I might also ask you to talk a little bit about what would you recommend for people in organizations that are just starting to move toward incorporating the decision-making criteria within the trial design and this framework? What kind of advice would you give to people who want to move in this direction, and maybe talk about a little bit about not only from the statistical standpoint, but what else their organization, their infrastructure needs to take into account before moving successfully into the approach that you described today?
Yeah. So the first part, the method that we've been using came to my attention in the company in, I think it was maybe second quarter 2012. And at that time, we were operating with kind of everybody writing their own code and doing their own spreadsheets and stuff like that. And then we kind of graduated to a unified set of tools, and then we kind of reached out to Cytel and started developing software there. And that's all that's the technical side of things. The other side of things that I've kind of touched on occasionally here is that there's a big cultural side of things to this. And one of the reasons why it was successful in AstraZeneca is that the value of having this procedure was shown to the upper management, people who are going to decide whether or not to fund your trials. They bought into it, and then they were able to kind of cascade the need for having this go-no-go criteria framework down to the study level and the project level and so forth. So what I would recommend, and I've had the privilege of seeing the spreadsheet full of everybody who's attending here, and I really appreciate all the interest. It's a much bigger audience than I thought I would have gotten. There are a few people at various levels. So for those people who actually might even sit on these governance boards, and I'm sure that other companies have them, communicating the method and the benefits to the other members of the board is certainly one thing I would recommend. And then in parallel, you want to try and spread this at the project level as much as possible, and even so much as to, for lack of a better description, familiarize or train other disciplines in your groups. So, in my opinion, the guys who really get the most out of this, aside from statisticians, are physicians, because they can, kind of get a sense of where things are going to wind up going at the end of the study and what's going to wind up being good enough or not good enough. So they wind up kind of increasing some of that quantitative orientation that they probably have, but it's not as solid. Solid's not the right word, but I mean it's not maybe not as in focus as we have it initially, and then through them, and even in parallel, the other disciplines, data management, clinical, etc., so that everybody kind of gets an idea that, when you're doing your trials, the very important information is not only going to be just kind of written up in a CSR somewhere, but it's going to be taken and used in a decision process by those in the company who are holding funds. I think that would go a long way to try and enroll people into the process.
Pat, thank you so much. That no doubt there was some great insight there. I just had to ask you. You mentioned the upper management and were persuaded. I think you said precisely they bought into it. Why? What do you think the selling point was? What was it that really moved them?
I think a couple of things. I mean, first off, with the dual target framework, and I think this is probably true of all of them, you wind up delivering trials that can be decisive at a lower sample size. So, you saw the quote earlier from Eric Polkstenis that a single criteria isn't going to do the job anymore. Now, I think he's right about that. I think that you're really trying to take something that's more on a continuous track here, even those that are discrete and so forth, and you're trying to write one line in the sand and say, okay, this is good and that's not. Well, I mean, if you've been doing this in a while, you've run into the situations where people want to take continuous variables and cut them into two categories, and you could lose all this information and so forth. We don't really do that here. So you have the sample size benefit. I think there's also a standardization benefit. So and I'm trying to imagine myself in the decision maker's shoes here, that if all my proposals come with a particular set of risks, then when I consider the factors of time and risk and cost, which usually are the big things, I think, that kind of they're project independent in consideration of a lot of these things. Having something that speaks a consistent language, that is something that maybe we can control a little bit through policy. We tell people, right, look, we want things that are decisive at a particular level. Then that kind of reduces the dimensionality a little bit. Okay, if I know that things are going to come with 30% alpha or 30% amber or lower, then I can then focus on time and cost, and know that things are going to work out more or less in the end. A lot of this is speculation on my part, I'm afraid. But I hope that some of that helps. that kind of helps us illustrate things, I guess.
Absolutely, Pat. You have Charles and I both nodding our heads in agreement. So I can't imagine better insight given your position, and I could see where the case could be rather convincing, starting with the very fact that the reduction of sample size and a greater, shall we say, flexibility with greater precision.
Yeah.
I think we're at the point now where we're going to wrap up. I see there's still many people still with us, half the hour, and we appreciate that. We appreciate your interest. Again, you can expect a follow-up by Monday, and you can also expect to see another invitation to a trial design innovation series of webinars with folks like Patrick again in the near future. And so until that time, I thank once again Charles Liu, and especially Patrick Mitchell of AstraZeneca, and all of you for joining us today. Good day to everyone.